Let's move on into our last lecture then of this unit, kind of bringing everything together that we've been talking about here the last couple of lectures with regards to respiration. We, talked about in that lecture of the respiratory system, there were 5 components of respiration. We talked about 4 of them. The 5th was, I told you guys, going to be discussed in this lecture, the use of oxygen and the use of food substrates, most notably we'll be talking about glucose, in order to produce energy in the form of ATP that powers things like our muscle cells when we're out for a bike ride. And in that process, we produce some byproducts, one of which we don't see or kind of, well, we can't see carbon dioxide either, I guess, but we produce heat. And so we'll talk about heat balance towards the end of the lecture, which is directly correlated to cellular metabolism. And then carbon dioxide, which we've been talking about, that we move into our lungs to be exhaled out in the opposite direction from our O2. Now, right away, I'm going to include a lecture slide here that we've seen before, just to refresh our minds on what metabolism is. This lecture slide was included in our chemistry lecture at the beginning of the semester. That was our second lecture of the semester. We learned that metabolism is a term that refers to a sum of all chemical reactions taking place in our body cells, or inside of us, in other words. 2 forms or phases of it exist: catabolism, or catabolic reactions, and then anabolism, or anabolic reactions. And they are the inverse of one another. And in that, one does one thing and the other one does the opposite of it. Catabolic reactions are those that break down complex organic molecules into simpler ones, like a dimer, which we could say like a disaccharide, into smaller, simpler ones like 2 monosaccharides or 2 monomers, like we show in the picture there. We just got done talking about digestion, and that was, an example of catabolic reactions. We're going to talk a lot about catabolic reactions here today with regards to glucose and how we break it down into simpler substances to extract the energy from it, okay, which is why we do it. These chemical reactions are exergonic, meaning they produce more energy than they consume, releasing the chemical energy stored in organic molecules, which is what I just said. This is why we perform catabolic reactions regarding glucose and other food substrates. We take the energy from those fuel nutrients and we extract the ATP from them. Okay, in other words, we break covalent bonds, producing smaller molecules and release useful energy. We also talked previously about hydrolysis reactions, and that's what's illustrated here in the picture, in the middle of the page. Chemical reactions that not only break down complex compounds, but also release the energy from them in the form of ATP. A hydrolysis infers that water is involved. Water is an input that then we use to help break the bonds involved. Anabolic reactions are those that combine simple molecules and monomers to form our body's complex structural and functional components, like when we bring in those amino acids from our digestive tract so that we can take those amino acids into like our muscles and make strong proteins with it, okay, because proteins are bigger than the individual amino acids we're building up. These chemical reactions are endergonic, meaning that they consume more energy than they produce. This energy is going to come in the form of ATP that we need to have for this reaction to occur. What's nice is that we form the ATP here to power this, demonstrating that these reactions are coupled. They go together. I'll get to that here in a second. Now, ATP can be formed 3 different ways: through substrate level, oxidative level, or oxidative, and what we call phosphorylation. Phosphorylation refers to the movement of a phosphate to a ADP, making ATP. Remember, ADP is adenosine diphosphate, where there's 2 phosphates, and then ATP is short for adenosine triphosphate, where there are 3 phosphates, okay? Humans have the capability of performing 2 of those ATP forming methods. Anybody have a guess as to which of these 3 we cannot do? the last one. That's what plants do. Plants perform, what's that process called? Photosynthesis. And they make their own food, but they also make their own energy from light energy as well as the carbon dioxide that's in the atmosphere. We don't have chloroplasts that can, make our own energy in that means. So we have to consume it by like eating those plants or eating, meat from an animal that did eat those plants. Okay. Opposite of hydrolysis is dehydration. Synthesis, what's shown down here, occurs when water is removed. So we pull out the water, which causes the smaller subunits to be fused together. As I said, those two reactions go together. Two forms of metabolism go together. We call that coupling. So when complex molecules are broken down by catabolic reactions, like what we show right here, going from larger complex to simpler, we form ATP. Not all of the energy that's extracted from those complex molecules can be transferred into the form of ATP, because a lot of energy is released in the form of heat. Think back into the beginning of the semester, we learned that heat is a form of kinetic energy. So sure, we transfer that energy from something complex to something simple and form ATP, but a lot of heat is given off as a byproduct, meaning that that's energy that can't be used to power our cells. Now, the great thing about our body is, and us as humans, we're mammals, which means that we are warm-blooded, so we don't just produce that heat and give it off and have no benefit from it. Heat gives us a body temperature that is nice and warm, that we then can maintain through our thermoregulatory mechanisms like sweating and shivering and that kind of stuff, which we'll talk more about at the end of the lecture. The opposite of that takes place when simple molecules are combined, like those amino acids or the parts of a triglyceride, and we form those, which requires ATP. ATP that we just formed through the catabolic reactions. Again, same thing, energy is released in the form of heat, and it's about 60%. So we're only about 40% efficient in the usage of ATP and the making of ATP, which means that we're not very efficient machines. Okay? A lot of our energy is given off as heat, but that's okay. We get enough energy in the ATP that we do form. Another example of a coupled reaction is shown here on this page. This is the first time we've talked about these types of reactions, but they're very important to understand and know here in the lecture. Because these are going to play a role throughout our discussion on cellular respiration. So let's read through. Two important aspects of energy transfer are oxidation and reduction reactions. Now, sometimes you'll see it listed as, instead of oxidation reduction reaction, they call them redox reactions. I actually have it listed here as redox, the very next sentence, just a shortened version of oxidation reduction. So these are important. And then mechanism of ATP production. They are the opposite of one another. One allows for the removal of electrons from something. Ooh, my figure's all messed up here. What the heck happened? That's what it's supposed to look like. Huh. Well, the point still remains. Is it on your page at least? You can see it fine? Okay. So what we're going to do, this is the chemical formula for cellular respiration of glucose. C6H12O6 is glucose. Okay, that's the chemical formula for glucose. We oxidize it. There should be a line coming up and then down to form carbon dioxide. What we're going to do is we're going to extract the energy from glucose, because anytime you oxidize, you decrease its potential energy. We pull out a lot of electrons, and with it come hydrogen atoms. which is why what we're left with is no hydrogen, but instead what we're left with is carbons and oxygens forming CO2. Okay. This is the CO2 that we are breathing off every second of every day. Okay. So I like analogies. Okay. I like to think of this kind of like a relationship that I grew up having with my siblings. Okay. I'm one of 4. So I had 2 older siblings, 1 younger sibling. I was in the middle. I was the 3rd child. Okay. As little kids, and I'm seeing this with my 2-year-old, 4-year-old, big brother does something, little sibling likes to do the same thing. Okay. It's inevitable where big brother goes, little brother's going to tag along. Okay. Well, electrons and hydrogens are kind of in that same relationship boat. Okay. Where electrons are like the big brother or big sister, if you're a female out there. And then the hydrogens are like the little sibling that's going to tag along. So when we snag the electrons from glucose, we're going to also carry along those hydrogens. Okay. And in order for them to be carried somewhere, we need some hydrogen electron carriers, which is what these are, NAD and FAD. Okay. These are called coenzymes. And it's not shown down here, but they carry those electrons that we pull from glucose, and they carry the hydrogens, the little sibling, to the mitochondria for the ultimate making of the ATP, which is the whole reason why we're doing this. Okay. NAD is short for something I'm not even going to read, a derivative of B vitamin niacin. When it picks up electrons, it also picks up a couple hydrogen atoms, and it becomes NADH plus H plus. Now, you'll sometimes see it listed as, and I'll show you in a couple of slides that it's listed this way. Instead of listing it this way, it's called NADH plus, or NADH2, and I'll show you why here on this page. Okay. Going from NAD to NADH plus H plus, as it's shown here, that second hydrogen is not directly connected to the molecule. So it's like, if we were thinking about these like as cars, carrying big brother and little brother, this little sibling is like, it's on top of the car, or it's in the trunk. It's there, but it's not directly connected. And so you just have to know that second hydrogen is there, but it's not directly connected. Now, with the case of FADH2, both hydrogens are connected, and FAD is what it begins as. When it gains the electrons and hydrogens, it becomes FADH2. FAD, same thing. I'm not going to read off what it's short for. You just need to know what is FAD. It's a derivative of the B12 vitamin riboflavin, demonstrating that not just glucose and carbohydrates and proteins and fats are good for a diet, but a well-balanced diet is important because it gives you your vitamins and minerals that also play very important roles in our body. So when we snag electrons and hydrogens from something, they're going to get carried somewhere and donated to somebody. So in other words, something's gaining electrons. We call it a reduction reaction. When something gains electrons, think of it, it's reducing its overall positive charge, because as we know, electrons are negatively charged. Okay? This increases the potential energy of the molecule, and who ends up getting reduced is oxygen's getting reduced, and it forms the water. So those electrons and hydrogens that were snagged from our glucose end up going somewhere. Oxygen accepts them, and that's why we have to breathe in oxygen to make ATP, at least aerobically, and that's what ends up forming water. We call this metabolic water, which is not a super, super important thing, because it's just not enough that we could survive without drinking water and getting it into our diet. And now, looking at this next page, we kind of show what takes place when those NADs and FADs gain those electrons or hydrogens, or I shouldn't say and hydrogens. NAD plus becomes reduced. So what you'll see these listed as is sometimes, in this form, NAD plus, this is its oxidized form. And you could say that for FAD. Might actually be a good idea down here in this little blank area Put oxidized for both NAD and FAD. That's its oxidized form, which means it's not containing the electrons that it does gain along with those hydrogens here and here. NADH plus H plus and FADH2 are referred to as what we call reduced coenzymes. So under here and here, it might not be a bad idea to write out that they are the reduced form. Reduced means, once again, they've gained electrons and hydrogens. They are the things that carry the electrons and hydrogens from that original glucose that we just talked about to a segment of our cell respiration called the electron transport chain. Probably have heard of that before. So let me read through. When NAD plus is reduced to NADH plus H plus or NADH2, the NAD plus gains a hydride ion. So the positive and the negative end up neutralizing each other. So that's why you see it listed as NADH without any positive or negative. The hydrogen ion that's not attached is still positively charged though, because it's out there, it's still an ion. When NADH is oxidized to NAD plus, the loss of the hydrogen ion results in one less hydrogen atom and additional positive charge. What this illustration really could show you is that you could draw a line or an arrow pointing back from NADH plus H plus to NAD plus. Same thing over there for FAD and FADH2. These are reversible reactions, meaning when we pick up these electrons and hydrogens, they're not stuck there forever. They can be given back to something else. Okay, like I said, I kind of think of these like vehicles that carry big brother, little brother from one place to another inside our cells. Okay. FAD in its oxidized form becomes reduced as FADH2 as it gains a hydrogen ion and a hydride. It's then oxidized to FAD when it loses those same two ions. Those are coupled reactions, meaning that each time one substance is oxidized, another is simultaneously reduced. Like I said, we call them oxidation or redox reactions for short. When something is oxidized, we describe it as exergonic, usually anyway, meaning it releases energy. This is what we once again do to our glucose. We're stealing its energy. The energy comes from the electrons in the glucose. That way we can transfer that energy eventually to form our ATP. What we show here in this figure is what we call the electron transport chain, where the electrons that we take from glucose, the hydrogens that we take from glucose ultimately end up. A series of proteins. Here we can show NADH plus H plus carrying those electrons. Here's an electron. There's its hydrogen. What we do is we give them away. So it goes from the reduced form back to the oxidized form, NAD plus. FADH2 does the same thing, becoming FAD. And the energy that's extracted from those electrons and hydrogens, it's not shown here, ultimately is what we then use to transfer and generate ATP. So we're going to see a lot of NAD plus, NADH plus H plus, FAD, FADH2 here in this lecture throughout the 4 stages of cell respiration. We're not quite ready to get to that point yet though. Now, glucose is a carbohydrate. We're going to spend the majority of our lecture talking about carbohydrate metabolism. There are other points of the lecture where we'll get into protein metabolism and fat metabolism, but the bulk of this lecture is dedicated to carbs. Now, cellular respiration that I've been referring to is the formation of ATP from glucose. Okay, and that's an example of a catabolic reaction. But catabolism, or catabolism, that's the way to pronounce it, is only one of the 2 forms of metabolism. Remember, there's building up, there's breaking down. Okay, So we'll actually talk about breaking down, but we'll also talk about building up with regards to carbohydrates. Before we can do anything with glucose and the other monosaccharides, fructose and galactose, we must carry those molecules into the body from our intestinal tract. What we just got done talking about in our last lecture, absorb them from the lumen of our small intestine, which is going to take place by a process called sodium cotransport, which is what this figure is showing. This would be the lumen of the small intestine. This would be one of those absorptive cells, the interior of it. We bring that glucose in, and it's going to take a piggyback ride with sodium to do so. We talked about that in the last lecture. Hopefully you watched the video, and that is a ring in the bell. Once we bring that glucose and the other monosaccharides into the blood, what we do with it is dependent upon the needs of our body cells. Okay. We can bring it into the cells to make ATP. That's cell respiration. That's what we're going to talk a lot about here in just a second. We can also use that glucose and make amino acids. We can make glycogen with it. There's one of those 6 buzzwords that I wrote down for you guys here not too long ago in our endocrine lecture. Glycogenesis is the formation of glycogen from glucose. If we eat a whole bunch of sugary stuff, we'll end up getting fat, right? It's because we can actually convert glucose to fats, triglycerides, a process called lipogenesis. But if we're going to store fat, if we're going to store glycogen, we have to bring those molecules into our cells, which is what this figure is showing you with what we call GLUT transporters. Glucose transporters are proteins that carry glucose from high concentration to low concentration. That's a form of facilitated fusion. Previously, we learned that insulin is secreted when our blood sugar is high, meaning that we want to get glucose out of the bloodstream into our cells. Insulin is going to promote the insertion of lots of these into our cell membranes so that we pull a lot of glucose out of the blood. Now, once that glucose enters the cell, it gets phosphorylated. That means it's going to add a phosphate to it, which means it's kind of trapped inside the cell. Then we can do what we need to do with it. We can use it for ATP production, or we can do those different things that I just mentioned. We'll kind of hang on and talk about those other things here in a little bit. Really quickly, though, I'm going to write out those 6 terms again for you guys, so you have a refresher on them. We're going to talk about each one of them in various points of the semester, or in this lecture. The first one, and this is the one we're going to talk about in great detail here in just 1 second, is glycolysis, which is the breakdown of glucose to form ATP. Two out of those 6 terms were on that slide. Let's look at one of those 6 here. Glycogenesis, which is glycogen formation from glucose molecules. This is what we do like when our blood sugar is really high. We store glycogen in our liver and our muscle cells especially. That way, when we need the glucose, we can release it by glycogenolysis, which is the exact opposite of glycogenesis. Glycogen breakdown, forming glucose molecules. That's what we do like when our blood sugar is really low. We also, when our blood sugar is really low, can perform what's called gluconeogenesis, which is the formation of glucose from non-carb sources, things like fats and proteins. Okay, so those are all various forms of metabolism relating to carbohydrates. We can also perform lipogenesis. That was something I just mentioned on that page. Fat or triglyceride specifically formation. And the opposite of that is lipolysis, fat, triglyceride breakdown. So these terms should be familiar to you by now. If they're not, make sure you know them. This is like the third time we've referenced them. We're going to get into them again here today. But we know about them and what hormones are involved with stimulating them. We'll refer back to those hormones again in this lecture. That'll be kind of more towards the end of the lecture. For now, we're going to focus in on glycolysis, which is a form, or which takes place as part of cellular respiration. So that's a component of this that we're going to move into right now. Cellular respiration is the process releasing useful energy from food in order to produce ATP. In other words, it's the oxidation. What's oxidation again? The removal of electrons and hydrogens from glucose. Ultimately, what we form is the main goal, ATP. But as we know, anytime we perform a chemical reaction, heat is a byproduct. Great thing is, and I've already mentioned this, the heat that we produce gives us a body temperature. Glucose contains a lot of potential energy. Potential energy is just the energy stored in the bonds of the glucose. When we break it down, we form carbon dioxide and water. Carbon dioxide is what we circulate to our lungs and exhale. In order to power this, not only do we need glucose, but we need oxygen. That's relating right back to what we just talked about. Once again, demonstrating why I talk about this lecture right after I talk about breathing and eating. Okay, we get our glucose from eating, we get our oxygen and remove our CO2 by breathing. That water I mentioned earlier that we form is called metabolic water. Like I said, it's not going to be sufficient enough that we can survive without drinking water, but it does help to meet our daily need for water. The energy in the form of heat, once again, gives us a body temperature. But more importantly, the ATP that's formed powers the different things that we've talked about this semester. We learned how important ATP was for like active transport, pumping stuff against its concentration gradient. How ATP is very involved with muscular contractions and muscular relaxations. How it creates the power stroke with myosin and actin. ATP is needed for cellular division and other various cellular processes. Now, in order to synthesize ATP, it means that we must bond a free phosphate molecule to ADP. Once again, ADP is adenosine diphosphate, 2 phosphates. If you add a third, that makes ATP adenosine triphosphate. Good thing is ADP and those free phosphates are very plentiful in our cells whenever ATP has been broken down for energy requiring processes. So when we use ATP, it's not like it's completely gone. What we're left with is ADP and PI, inorganic phosphate, free phosphates. We can then just put it back together and make ATP from it. I just summarized cell respiration of glucose very simply. As it says there, it's not quite that simple though. It's a complex series of equations where we take glucose apart piece by piece, removing hydrogens and electrons and splitting carbon to carbon bonds, releasing energy of glucose gradually, so a significant portion, 40% or so, is available to synthesize ATP. For a kind of more detailed illustration of what's taking place, there are 4 steps involved. Got a little bit of a background on each of them and a picture to show them where they're taking place with regards to our cell. Begins with that first term I wrote down on the piece of paper, glycolysis. A set of 10 reactions that take glucose and split it into 2 what we call pyruvic acid molecules. In doing so, we do form a little bit of ATP. We extract electrons and hydrogens, forming NADH plus H plus, which we're going to do at various other points in this process, like step 2. Step 2, the textbook listed out as formation of acetyl coenzyme A. I typically call it the transition reaction because it takes place between glucose breakdown, glycolysis, and the Krebs cycle. It's kind of a intermediate step. In fact, a lot of textbooks actually just include the transition reaction as part of the Krebs cycle because they happen bang, bang right after each other. There's no ATP formed in the transition reaction, but we do produce some CO2. That's CO2 that is breathed off. We also form that during the Krebs cycle. We do also oxidize the acetyl-CoA, or the pyruvic acid, which is helping to form acetyl-CoA, and that forms NADH plus H plus. We form a lot of NADH plus H plus and a little bit of FADH2 in the Krebs cycle, along with a little bit of ATP. Now, both the Krebs cycle and the transition reaction are taking place inside of this, what we call our mitochondria. Remember, mitochondria are described as our powerhouse of the cell. And this is where the majority of our energy is formed, but not so much here, but step 4, the electron transport chain, where the majority of our ATP is formed in a process called oxidative phosphorylation, which tells you oxygen is involved, hence the name oxidative. And we also form a little bit of water called metabolic water that I've talked about. Okay, so I just kind of have set the stage for what we're going to start with right now with glycolysis, where we split a molecule of glucose into 2 molecules of pyruvic acid. We generate a little bit of energy in the form of ATP, and we oxidize it, forming some NADH plus H pluses. Okay, a little bit of information here on this page, and I'll kind of bounce back and forth between this page and the next page where I have an illustration to kind of really make some sense of it. Okay, here's the figure I really like to use. It's a figure actually I snagged from the biology textbook I used to teach when I got to teach biology. It's a lot simpler of an illustration than what your textbook provides. Your textbook gives you the more detailed biochemical process and vision of what's taking place. It's a series of 10 steps, 10 reactions. You can see they're all numbered here. But I kind of just like to use this illustration to kind of break it down into kind of 2 phases. What we call an energy investment phase and an energy harvest phase. Okay. Now thinking back to glucose, one of our inputs, glucose we said earlier is C6H12O6, meaning it has 6 carbons, 12 hydrogens, 6 oxygens. Okay, let's just focus in on the carbons right now. C6, 6 carbons. Each of these little gray dots in the figure represents the number of carbons it has. So, or represents a singular carbon. So there are 6 here. There's our 6 carbons for glucose. Okay. Another energy input, we call this the energy of activation, is 2 ATP. This is what starts the process. That's why we call it energy of activation. It's activating the process. It makes glucose unstable, and it's basically going to break it in half. Because what happens is a phosphate from each of those ATPs is transferred to the glucose. And through some kind of different things, the glucose becomes unstable and it breaks into 2, 3 carbon molecules. 1, 2, 3, 1, 2, 3. Okay. Now, what we do next is we extract some energy in the form of oxidizing it. Oxidizing means that we extract electrons and hydrogens. And that's taking place right here with our NAD forming NADH, NAD forming NADH. Now, remember, there's a second hydrogen, so whether you write it as NADH plus H plus or NADH2 like I do right here, okay, it might not be a bad idea to kind of keep your head straight. You know, right next here, put plus H plus. Down here, put plus H plus. Because there's another hydrogen there. I just don't know why they don't include it in the figure. Okay. Now, these things are going to come into play later on, into the final step of making ATP. We have 2 ADPs and, 4 ADPs and 2 phosphates coming in, 2 here and 2 here, where we can actually form a little bit of ATP. 2 here, 2 here, 2 plus 2 is 4, right? 4 ATP is listed here as our output, but notice here in parentheses I listed as a net of only 2. Why do I say a net? Because it's not like you started at 0. You started actually at -2. And I use another analogy for this. You guys know I love analogies. Okay. Whether you've ever been to a casino, I know you guys are probably young, haven't, but you all know how a slot machine works, I assume. You put some coins in the machine, you pull the lever. Okay. In order to pull the lever and make anything happen, you have to put some money in. In order for any of this to happen, you have to put some energy in. So it's like walking up to the slot machine and putting 2 quarters in, pulling the lever, 4 quarters pop out. How much money do you truly win? Not 4, but 2. And your net total winning is 2, even though you've got 4 in your hand because you put 2 in the machine to start the process. So that's why it's 2 and a net of 2 here, because you have to subtract these 2 from the total 4. And what you're left with are 2 3-carbon molecules we either will call pyruvic acid or pyruvate. Kind of can be used interchangeably. Okay. And keep in mind, this is all taking place in the cytoplasm. So not inside the powerhouse, not inside the mitochondria. And it's anaerobic in nature, which we learned early in the semester means does not require oxygen. Let's see, let's try this video. What the heck? That's okay. Here's the same video. I don't know. That link must have been broken.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream.
Okay, so what they were just showing was the entry of glucose into the bloodstream from our intestines. So that was related to what we talked about in the last lecture.
From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase,
Okay, right here they're showing NAD plus coming in, and it's going to snag some electrons and some hydrogens, forming NADH plus H plus. So I paused it because she kind of just skips right over it, but watch it take place. I'll back it up 1 second.
the phase begins. During this phase, the two 3-carbon
There they go.
molecules
There's the
are
electrons and
converted to
hydrogens going
pyruvate,
away.
and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is
Okay, so she's starting to lead into what takes place next. So I'm going to pause the video and pick back up where we were at.